So welcome everyone to the May webinar of PASTA, the Paleoproteomics and Archaeology Society for Techniques and Advances. The purpose of these webinars is to inspire connection and communication between members of our community. So please feel free to ask our speaker questions at the end of the talk. However, these webinars may contain published or unpublished data, and resulting conversations and content should not be shared without the express permission of the speaker. Please note that these webinars are not formal presentations necessarily, and the speaker should be contacted directly if you are seeking interviews or statements for reporting. And finally, we want to encourage a safe and positive space for the sharing of knowledge and information in our community. And we ask everyone to abide by our code of conduct, which I've posted into the chat. So this week, we are really excited to welcome Claire Koenig from the Globe Institute, who will be talking about automated high-throughput biological sex identification from archaeological human dental enamel using targeted proteomics. So with that, I will pass it over to Claire.
Thanks a lot, and thanks for inviting me. And I'm very happy to be here today to talk to you about biological sex identification using proteomics. So the work that I will present today is based on this preprint that recently got out, and that has a very long title, the automated high-throughput biological sex identification from archaeological human dental enamel using targeted proteomics. And so to start with, I'm going to go over how we can perform sex identification using proteomics based on dental enamel. So here we have a teeth, and the enamel part is the outer layer of the teeth that is the most mineralized and the hardest part of the whole human body. And with that, it is very suited for protein preservation and therefore making it a very nice matrix for paleoproteomics. So this enamel matrix is composed of only a few sets of proteins. So we have mostly amelogenin, ameloblastin, and amelin, and other proteins in lower abundance. And what is very interesting in enamel is that amelogenin exists in two isoforms. So one that is encoded on the X chromosome and one that is encoded on the Y chromosome. So it means that in female individuals, basically both chromosomes are going to be X pairs, and so they're only gonna so we're only gonna find amel X protein in the enamel. However, whenever we have a male individual, we are going to be able to find both amel X and amel Y proteins, and that's how we perform sex identification. So with the only detection of amel X, we have a female individual. However, with the detection of both amel X and amel Y, we have the identification of a male individual. And at the moment, it's not something that is new. So there has been many papers discussing it and developing the methodology and optimizing the sample preparation on that, and also applying it in a very diverse context, very diverse time periods with different methodologies. So the question is, why do we need new methods? And that's exactly what I've worked on, developing new methods for sex identification. So at the moment, proteomics is very useful for sex identification as it's biological information. So it means that we are able to get like a very precise answer on the sex of the individual, and it does not rely on morphological features. It also does not rely on contextual evidence. So whenever a remain is found with the presence of hunter artifacts, then it will mostly lead to the identification of a male. However, with biological information, we don't have an identification that is based on our judgment. Also, it can be performed in a minimally invasive way, especially with acid etching. It's something that is very precise and sensitive as it relies on mass spectrometry. And mass spectrometry can detect a very low amount of peptides, and we can get a very nice and precise signal from that. And additionally, it can be performed on degraded samples. So unlike histological sex estimation, we do not need to obtain, like to have a full skeleton to perform sex identification. Only teeth or teeth fragments are required. And therefore, sex identification using proteomics is actually quite interesting, especially for archaeological research. However, it's very expensive. As I've said before, it relies on mass spectrometry, and mass spectrometers are extremely expensive, so the use of those makes the whole cost of the analysis quite high. Also, the complexity of the mass spectrometry-based proteomics workflow is quite high. So in terms of handling the instrument, but also in terms of data analysis afterwards and being able to interpret what we have. And it's also extremely time consuming. So it's not like an infrared technique where in a few seconds we have a readout. Usually the analysis of one sample will take at least 1 hour, and that excludes the data analysis that comes afterwards. And so all of this is limiting the analysis of to a few high-profile individuals. Additionally, using proteomics for sex identification, there is always an ambiguity in female identification, as whenever we have a female, we only detect amel X, but the absence of amel Y is something that is extremely hard to prove. And so now the main question that I ask myself is how to overcome those challenges. And to overcome those challenges, we set different objectives. So the first one is to have a cost-efficient method for sex identification. And the answer to that is to make the whole analysis as fast as possible. So that includes reducing the length of the sample preparation, the MS acquisition time, and also the search time that comes afterwards. The second objective is to obtain confident female identification. And to overcome this challenge, we are using a quantitative approach to model intensities so that we can obtain with high confidence positive female identifications. And the third objective is to make the whole process as hands-free as possible. And for that, we developed an automated data analysis pipeline with a user interface. And basically, this is the outline of my presentation. So I will start with the first part, that is having a cost-efficient method for sex identification. So right now, the workflow that has been developed consists of a very easy sample preparation with an acid etching and direct loading of the peptides into evotips with a fast acquisition, a fast separation, and a targeted data acquisition with, at the end, an automated data analysis pipeline. So here we will focus on the first part, that is the sampling. So using acid etching, basically, we can perform minimally invasive sampling. And then the peptides that are going to be in the acid can be directly dissolved on evotips that are directly loaded into the SC system. And in order to do so, I've sampled many teeth, and I found a strategy that worked well for me, and that was proceeding by batches. So basically, I was working by batches of 6, and I was putting acid in the lid of the tube and displaying my teeth that could either hold by themselves or with a little help. And with the time I needed to process 1 sample, then I would have 6 at a time, and therefore increasing the throughput of my sampling strategy. So the second part that had to be optimized was the LCM-SMS method. So first, reducing the length of the gradient, because the gradient is the time that is going to drive the whole analysis time. And in this study, we have a gradient length of 5.4 minutes. That corresponds to the analysis of 200 samples per day. And I've compared this gradient length to different literature performing sex identification that usually range from 1 hour to 2 hours. So here we could have quite some drastic decrease. There's also this paper that performed direct injection, and in that case, the analysis was extremely fast as well. By reducing the length of the gradient, we can also increase the throughput of our method. So here, I've compared the number of archaeological samples analyzed in our study, so 339, to previously published studies. And I mean, the second study that analyzed the highest number of archaeological samples was 75. So by drastically reducing the length of the gradient, you could also like clearly increase the throughput and analyze many more samples. So the second part here is focusing on the MS method. And what we need to do is to change the way we acquire proteomics data. So here, if you take the example of a classical chromatogram, so we have the signal across the time that is on the x-axis. If we take a given time on that chromatogram, in a data-dependent acquisition scheme, what is going to happen is that we're going to acquire a full scan. On that full scan, every peak is going to represent a peptide ion. And those peptide ions are going to be selected and isolated for fragmentation. So the first one is going to be selected, isolated, fragmented, and the tandem mass spectra is going to be acquired. The same for the second one, the third one, and the fourth one. And here it's an example of a top 4 method, but basically afterwards, we get a full scan again and different tandem mass spectra. However, we decided to use a different type of acquisition scheme that is called PRM, for parallel reaction monitoring. And in PRM, what is very important is here our inclusion list. So we need to have a list of masses that the mass spectrometer is going to target. And if we take a certain time t in our chromatogram, we will generate a full scan. In that full scan, the mass spectrometer is going to look for the masses that are included in the inclusion list. So here we can see the third one and the seventh one. So the third one is going to be selected, isolated, fragmented, and the tandem mass spectra will be acquired. Then the second one. However, there's no more peaks that are in our inclusion list, so there will be a full scan again, and the cycle is over. In the case where there's no peak that matches any mass on the inclusion list, then a full scan will be generated again until a mass is found. So in DDA, that is the most used acquisition strategy, especially in paleoproteomics, we obtain the measurement of a comprehensive proteome. However, in PRM, we have a precise quantification measurement over a limited number of targets. So it means that here we can perform precise quantification, but if we have a peptide that is not in our inclusion list, we will never find it. And here I want to highlight the importance of actually picking the right targets for building the assay. And to pick the right targets, we did an analysis starting from 6 female teeth and 5 male teeth that were analyzed using a longer gradient. Here it's 44 minutes with a DDA acquisition scheme, and the data was searched using MaxQuant. So here we looked at the site intensity of AMEL-X and AMEL-Y. On the top, we have every site that is differential between AMEL-X and AMEL-Y. And what we wanted to look at are the sites that are the most abundant in terms of intensity, and that are differential between AMEL-X and AMEL-Y. And so we could identify the position 5965 and the position 196 to 102. After that, we performed additional filtering, as we were looking at sites, but what we detect in the mass spectrometer are peptides. So we filtered based on the peptide length in order to not have too long peptides, based on modifications, as we do not want to have peptide modified. We didn't want any isomers. We wanted a symmetric elution profile and also high intensity. So finally, our choice of targets are focused around the position 59 and 65, and we have 2 targets for AMEL-X and 3 targets for AMEL-Y. So what we can observe here is the MS traces at the MS2 level. So basically, we have the retention time on the x-axis and the intensity of the y-axis, and every different color represents a fragment ion. So it's every fragment that they use across the elution profile, across the chromatographic gradient, and the quantification is performed by calculating the area under the curve. So we benchmarked our method by using 12 replicates that we ran, and we looked at the coefficient of variation across the replicates per target. And overall, we have a variation that is below 7%, and in that case, we can say that our method is very precise. So the next step that we wanted to shorten was the search time. So in a classical DDA scheme, we would use maxQuant, and we would search the data against a protein database. And I guess many of you have already used maxQuant. The processing time is quite long, especially if you have many modifications or if you have unspecific cleavages. In our case, we did not use maxQuant as we have a different acquisition scheme. What we used is a software Skyline that is not performing a database search, but extracting the eigensignal directly from the raw data. And in that case, the time it takes to load our files in Skyline is a few seconds per file. I mean, it's very fast, especially here with short gradients and a limited number of targets that are 5 in our case. And so altogether, we could shorten the whole pipeline, so including the sample preparation, MS acquisition time, and search time, to have a method that is as cost efficient as possible. So then the second objective was to obtain confident female identifications. And to do so, what we rely on is having a very precise and accurate quantification of our targets. And basically, we mostly rely on the fact that we have a linear behavior between AML-Y and AML-X intensities. So here we have different male individuals that were plotted, and we do have a linear relationship. So still, there is some variation, and we wanted to see where this variation comes from. So, as I've already showed before, our methods show very high precision, so the variation that is observed cannot come from measuring errors. Additionally, we took 2 different male individuals and we performed a dilution theory. So with this dilution theory, you can see that we have very high linearity. So altogether, we concluded that the variation that is observed here comes from inter-individual variation. The way we perform sex identification is based on this relationship. So the first step is to calculate the limit of detection and quantification of all of our targets to then infer the limit of detection and quantification of both AML-X and AML-Y. So to do so, prior to running any sample, we need to run standard dilutions of all the different targets, and based on that, we will calculate the limit of detection and quantification. Then, for every sample that has an AML-X and an AML-Y signal, we will report all the points on our plot, and we will generate a linear regression in order to build a model. So whenever we have a sample that only shows AML-X intensity but no AML-Y intensity, then we will refer to our model in order to calculate a predicted AML-Y intensity within a 95% confidence interval. So whenever we have our predicted AML-Y intensity that is above our limit of quantification, but also the lower range of our confidence interval that is above the limit of detection, then we will have a female individual. In the case where the intensity basically the predicted intensity, do not fit our criteria, we will have a non-conclusive sample. And we decided to add this third option as to not force female identifications and to add basically a non-conclusive analysis in case the intensity was too low. So once we developed the model, we had to validate it. So for that, we used samples from the Ratton-Perez collection. So we had 10 males and 10 females that we sampled. They were analyzed through the whole pipeline. And the issue that we had is that we could identify 9 males and 11 females. And actually, this sample 14 that was originally a male, that we identified as female, that was a bit problematic. So we decided to investigate further on this one, because it could have been identified as a female if something went wrong with the analysis, and especially the data analysis. So if some signal would be misintegrated. So we looked back at the MS traces, and we can see that we actually only have noise for AML-Y, but we have clean signal for AML-X. So it just shows that it doesn't come from our data analysis. Then the next question was, were the target peptides absent from the sample? So it means that there was some AML-Y, but not just the target peptides that we looked at. For that, we performed a DDI analysis where we analyzed our individual 14, that was a problematic one, along with 3 other male individuals. And we can see that we have very similar AML-X coverage for all the 4 males. And we also can find AML-Y coverage on M1, M16, and M18. However, M14 did not show any AML-Y coverage. And in that case, we can say that it's not because of the choice of our targets that we missed, that we misinterpreted this individual as a female. And then what we asked ourselves was, okay, maybe the intensity was too low, but here the AML-X intensity of the individual 14 was the highest. So we would have expected, if AML-Y was present, to have some coverage in our DDI analysis. So our possible explanation is that we can have an AML-Y deletion, and in that case, we would be fully blind towards it, or the sample could also have been mislabeled. Altogether, we managed to validate our assay within a 95% confidence. So the next step is to make the whole process as hands-free as possible. And for that, we developed an automated data analysis pipeline and a user interface. So the way our data analysis works is that we take the raw data directly from our mass spectrometer, and we load this raw data into the software Skyline with a predefined template. So it takes a few seconds to a few minutes. It depends on how many files you have. Then we need to perform manual verification of the data, because sometimes the peak picking algorithm in Skyline can make mistakes. And here, for example, we can see the vertical lines represent the integration of the peak. And we can see that this peak is misintegrated. So manually, we need to reintegrate the peak properly in order to have a proper quantitative signal. And then we need to export 2 predefined reports that we will then load into a Shiny app. So the way the Shiny app works is that we need to load the reports, and all the signal is going to be filtered based on quality control from Skyline. Then the limit of quantification and detection per target and per experiment will be calculated, and all the signal that are below the limit of quantification are going to be filtered out. As we are performing quantification, we cannot have signal below that limit. Then other samples that present AML-Y intensity that is not 0 are going to be identified as males. Based on those male identification, a model is going to be generated within a 95% confidence interval. And there is also going to be an algorithm that is going to check for outliers. So there is a possibility of removing those outliers or not removing those outliers, and that I will show you later. Then the predicted AML-Y intensities are going to be calculated for the potential females. And based on the criteria that I've described before, we can obtain female identification or non-conclusive results. Then all the data is going to be combined into tables that can be exported with all the results. So the application is freely available on GitHub, and I will, before describing the different applications that we did, I will show you how the whole process is working. So here we have a Skyline session that where I have loaded my file. So here we can see we have just different samples. We have the different targets on the side. And what we need to do, once we loaded the file, we need to just make sure that the integration is taking the full peak. Then we need in the document grid to check our report because we need to give some information. So all those columns will need to be filled. So here we have our standards that needs to be labeled as standards. Then all of our samples need to be labeled as unknown. And if ever we have blanks, they also need to be labeled as blanks. The concentration of the standards needs to be filled and also the concentration multiplier, but everything is detailed in a tutorial on GitHub. So once we close, it is saved, and then we need to export the reports. So here it's peptide intensity data and raw intensity data that are custom made. So once we have our report, we need to generate a third report that is called experimental design. And so in this experimental design, what we need to do is to have a column that is called file name and a column called experiment. So it's a TXT file where we have all of our raw files in one column and a random name. So here I called it 1 as I only have one experiment. And then we can open R with the app and run the app. So sometimes it might take a bit of time to load the packages. Yes. And then we have a window that opens, and that's the window we're working with. So on the side here, we need to upload our data. So we need to upload one report that is called peptide intensity data that I load here. Then we need to load our experimental design table, and we can plot our standard curves. So here it's mostly a way to check that our standard curves are actually quite linear and that we can use it for quantification. Then we can go in the second tab and we can plot the model. So we will have a summary graph of all the males, all the females that were identified, and also the model that is used to perform the sex identification. So there's two options here. We can use the experimental model that is going to generate the model on our own data set, or we can use a predefined model that was generated on modern humans here in the lab. That looks like this. Then we can also plot the table where we have our samples, our MLX intensity, MLY intensity, the sex, and also the experiment. And this table can be downloaded as a CSV. And finally, the last part, we need to load the raw intensity data. And that's a part just to be able to check your data without having to go back to Skyline. So this table is a bit bigger, so it takes a bit longer to load. So we just need to wait for a few seconds. And here we can select the sample we want to look at. So we have every different sample and the blanks. So this is why it's important to define blanks as blank in Skyline, because if you define them as samples, they will be processed as samples. If you define them as blank, they will not be used for the sex identification, but they will still be visible here. So if you look at the first one, we can plot the tick, and we can see all the signal for the different targets and the sex identification here. So we have MLX on the top and MLY on the bottom. Then I also wanted to show you an example on another data set that we analyzed, because sometimes it can happen that you have some samples from one cohort that you want to analyze with their own standard line, and then a bit later you get like a follow-up batch that you want to analyze. But in this case, you would have to run another standard line for proper quantitative analysis, just because the limit of detection and quantification are going to be dependent on your instrument setup. And so in that case, what you can do is specify different experiments. So here, for example, in this experimental design table, I analyze my files in 2 batches. So I have my first standard line that is named experiment 1, all my samples. And then when I go down, I have my second batch, so with my standard line, and below my samples that is named experimental design 2. And the thing is that the script is able to handle this. So it doesn't have to be 2 different experiments. It can be more than 2. So I'm just going to change the experiment. And in that case, then we can see both standard curves. For generating the model, it's going to take into account all the males individuals, not just from one experiment, but from both experiments. And so here we are generating the experimental model, and we can see that we have some outliers. So we can actually remove those outliers by allowing the script to remove about here 10% of outliers. And if you plot the model again, then we can see that it got refined. We can allow up to 50% of outliers to be removed, but as we are going to see, we don't actually lose many points because it's either the outliers are removed until there's no outliers left, or the maximum amount of outliers allowed is reached. And then we can, again, plot the table, download the table, and have all of our sex identification. So we try to make it as user-friendly as possible. And yes, it's available on GitHub. So right now we have optimized the full pipeline, and we have applied this pipeline on archaeological samples. So the first set we applied it to was from Jordan, in that site that is in the greater Petra area. So we had 24 samples that were dated from about 9,000 before present and showed quite poor dental preservation. So we mostly had some teeth fragments that were run using DDA and PRM. And the main question was to check whether we can identify all the target peptides in ancient and degraded samples. So it was a bit of a stress test for our method. And what we can see is that in other samples, so for AMLX, that are the 2 first boxes, we could identify the targets. So in other samples. For AMLY, we could identify the targets in other potential male individuals. So we compared our results with our assay with DDA results, and we could see that we had a perfect correlation except for this individual, SM21, that could not be identified using the DDA assay. Some osteological sex estimation could have been done on a few samples, and we can see that the proteomics data is also correlating with osteological sex estimation. So based on that, we could conclude that our assay can actually be applied on archaeological sample. And in order to demonstrate the throughput of the method, we tried our assay on samples that were originating from St. Pölten in Austria. And so in total, we had 315 samples. And they're coming from the Domplatz of St. Pölten, that is the main square, so that you can see on the picture here. And so all this building is a darker area around. And they started by doing some excavation before construction, and they actually found a lot. So at the end, the excavation lasted more than 10 years, and in total, they managed to recover about 20,000 individuals. So we did not analyze 20,000, but 315 that were dated from 1359 to 1360, and they are thought to be deaths that are linked to the first wave of Black Death in Austria. And so we had samples corresponding to those mass graves, so SG2, SG5, and also 106 samples from smaller mass graves and a few skulls that were kept for anatomical studies. So altogether, we managed to analyze all of our samples and get a sex identification for all of them except one that was injected into low abundance. So we could get the sex of other individuals, and the thing that is quite interesting is that we could get an overview of the sex distribution in those graves, and we could obtain that in less than 2 days of mass spectrometric time with a very easy data analysis process. So basically making the study of population possible here. So altogether, we developed an end-to-end pipeline for sex identification using proteomics, and we demonstrated its applicability on archaeological samples, where we could showcase an unprecedented throughput with a tenfold increase compared to published method. And this tenfold increase is calculated based on the MS time. It doesn't take into account the sampling time or the data analysis time. And we additionally developed a semi-automated data analysis pipeline with a user interface for fast, reproducible analysis of the data and confident female identification. So with that, I would like to thank you all for your attention. I also would like to thank Push for founding this project, other members from my group, and other co-authors on this project. Thank you.
Really, really cool work. Thank you so much
Thank you.
for your talk and for developing such a fantastic looking data analysis pipeline for something that usually, I mean, at least what I've done, it's been manual and it's taken forever and been very annoying. So I'm very excited to try this out and we'll be emailing you with many questions, I'm sure.
No worries.
If anyone has any questions for Claire, they can post them in chat or can just unmute themselves and ask. I might just start us off because I have a question already. I was just thinking because you mentioned during your talk about individual variation between the peptides you were seeing, and I was just wondering how you were, if you were seeing kind of these bigger data sets you were looking at, if you were seeing kind of more of this individual variation and also intersite variation coming up in the different peptides you were seeing, or no? Yeah.
Actually, I did not really assess it, but I mean, in the model, I was not having more outliers using archaeological sample or modern human sample, but of course, the more sample we have, the more likely we are going to have outliers.
Yeah, no, that's totally fair. Thank you. I know your models were incredibly consistent. It was really impressive. Um, I think we have a question in the chat from Charlotte. I can just read it for the sake of the recording. You mentioned you have a probability-based processing to more confidently assign samples as female in the absence of amelogenin Y. How often do you see cases where amelogenin Y has not been identified, but it doesn't pass this probability test, and therefore you can't confidently assign a sample as female? And have you ever had cases where you don't see one of the amelogenin X peptides that you've actually targeted?
Okay, let me just read the question again. So I have quite rarely seen cases where AMEL-Y has not been identified, but it doesn't pass this probability test. Okay, I'm not sure I get the question right, but basically, usually in my experience, when there's MLY, we have a nice enough signal so that it passes the limit of detection and quantification test, and also the other statistical tests that Skyline is doing in order to check for the proportion of the different fragments and also the precursors. Um, so if it doesn't pass it, usually it's because the abundance, so the intensity is too low, and in that case, it's possible to load more in the mass spectrometer. Um, So if I ever had a case where I don't see the MLX peptide that I have targeted, so it happened to me a few times, but it's also on TIFF where I had a bit of a doubt in terms of sampling, just because, yeah, that seemed to have some kind of coating, and I was not sure I was actually getting enamel, and I was having no signal in the mass spectrometer, so I think it was a sampling issue. So I hope I'm answering your question.
Does anyone else have any questions or thoughts?
I actually just get a question from what you just said.
Yes.
Often in the past they used to coat archaeological elements with collagen-based glues to preserve it. How do you think that would work with your method? Will you have issues if there's a collagen coating on it, or will you still be able to detect the peptides?
No, I think that might be an issue. Um, so I would for sure recommend to remove any coating before doing analysis, and also to clean the teeth in order to not have dirt and bacteria and everything. But if it's a collagen coating, I mean the collagen proteins are quite long and it's proteins, but what we extract are peptides. So yeah, we might have some carryover on the column if they use from the evotip. I mean, overall, I would not recommend it. But I think with a PRM assay, we would be more prone to actually detecting our target peptides rather than with a DDA method, because we are actively looking for the targets.
That's good. So then there's hope if there is a collagen coating on it that you were not aware of that was
Yes.
there. It's awesome. Also, great talk, and this is such an exciting study. So thank you.
Thank you.
I have potentially a silly question. But you know, obviously this is you've gotten to this quite high throughput, even with the LCMS work. Have you consider like thought maybe you might try like something like Maldi, or is it just you're just going to get a crazy number of peaks that you're just going to have no idea what they are, and that's that kind of thought process?
Um, so the thing, no, I mean, I actually did not consider Mildy tough, but the thing is that with Mildy tough, it's mostly fingerprinting, so MS1 only. That I think could also work, but we would not get like a proper and precise quantification as we do with LC separation. What I considered, however, was to try with triple quadruple instrument, or instruments that do not have an Orbitrap that can also be cheaper. But yeah, I mean, we don't have those in the lab, so I could not try it.
Fair enough. Next steps, maybe.
Yeah.
Okay. Well, if no one else has any questions, um, speak now or forever hold your peace. But we'll just thank Claire once again for an amazing talk and a really exciting study. Again, if you have any questions, I'm sure that Claire would be happy to answer them over email potentially. But thank you everyone for coming to our May webinar. I'll go ahead and stop the recording now and see you all next month.